In this video, I'm going to describe how to handle data that has been obtained during stop flow setup of Q Box RP1LP. This data was obtained with two small insects placed in a flow through variable volume chamber that I demonstrated in earlier videos. So as I mentioned before, there are some parameters that have to be set up before data is collected, and that's flush time and stop time. When measuring small or inactive samples like these two insects, stop time should be long enough to get a good measurable CO2 peak, which you can see on this graph, and measurable oxygen dips. The CO2 peaks should not reach 2000 ppms because the range of the CO2 analyzer is only up to 2000 ppms, and oxygen dips should be at least 200 parts per million, as the resolution of the oxygen analyzer is about 40 to 60 parts per million. So you need to be able to measure something that's at least 200 parts per million, and 200 parts per million is 0.02% of oxygen. So these dips that you see here, are sufficient for stop flow. So we recommend that a short test run with your animal is performed like we did here, with about 3 to 4 cycles. And this is from this data one can figure out the best flush time, stop time, and flow rate through the chamber to get sufficient differentials. The differentials for CO2 and O2 are graphed in this graph, and you can see that we're dealing with about 500 ppm, both CO2 peaks and oxygen dips. When performing short test run, it's important, as I said, to set up the flow rate. In this case, the flow rate was close to 100 mils a minute. The flow rate needs to be fast enough to flush the animal chamber a few times during the flush time period. And you can see in this example that this flow rate of about 117 mils a minute flushed the chamber in about 1 minute. You can zoom in here on this scale so you can see one of the peaks more clearly. So I'll set this up at 10 minutes and 15 minutes. And the PWM, which is our indicator of whether you're in flush or stop flow, is graphed on the right side of this graph. When PWM is 1, the sample is in stop flow, so no gas is going through the chamber, and therefore we're measuring the reference levels. And when the PWM is 0, that's the flush period. That's when the gas is flushing through the chamber. And you can see that approximately starting here, the peak CO2 has been flushed out, and the dip of oxygen has gone through the analyzer in about 1 minute. I highlighted this portion of the graph. Delta X, which is our change in time, is about 0.8 of a minute. So what we want to see is a clear CO2 peak followed by a steady state. You can see that here in raw data, a steady state level before there's a switch from the flush back to stop flow mode, and the reference is being measured. The flow rate through this chamber will depend on things like the volume of the chamber, the length of the tubing between the chamber and the analyzers. The larger the volume, the higher the flow rate, or the longer the flush time, to make sure that all the gas is flushed and measured. So looking at the trial test data here, what we want to see is these sharp CO2 peaks, nice sharp oxygen dips, and then a steady state that follows before there's a switch to stop flow. So with this current data, the most appropriate analysis is stop flow. And I will go now through the setup of the other parameters, because there's 4 other user parameters which have to be set up before the full experiment is run, but they may be set up after the experiment is finished and the data is collected. These parameters are event time, O2 delay, delay high, and delay low. These user parameters do not have to be set up at the beginning of the experiment. They can be set up when the data is already collected. However, after you run this test experiment here, we have enough information to get these parameters fairly close to where they should be. So I'm going to explain on how to do this. To set these parameters, we're looking at the diff CO2 and diff O2 graph, which is showing the differentials. It's good to zoom in, just like I did over here, so you can see one of the peaks, very clearly. And the first parameter that we're going to look at is the O2 delay. The O2 delay is a parameter that lines up the CO2 and oxygen peaks. This is important for data analysis and calculations because the two have to be in alignment, and they may be not in alignment as a result of a delay between the two analyzers. The oxygen analyzer is placed after the CO2 analyzer, and depending on the length of tubing in between and depending on the flow rate, there may be a delay. So to line these up, what you need to do is we need to go into examine in the top menu, and what you'll see is this window pop up and the line. This line here is going to show us how well the two peaks are aligned. You can stretch this graph as well by pulling at a corner so you can see the data even more clearly. Bring the line to the top of the CO2 peak and see by how many points is the oxygen peak misaligned. And it looks like it may be about 2 or 3 points. So the next thing to do is you go to O2 delay, and you can either increase the delay here by clicking on this arrow and see how that changes the alignment, or you can double click on the window of that user parameter and you can type the number here directly. In this case, let's try 2 and see how that changes, and bring the examine line back onto the peaks, and you can see that the two peaks are now aligned. So with that said, we can close this window here, and then the next parameter that we want to adjust is going to be the delay high and the delay low. These two parameters determine where the software takes the data for integration of the peaks. So to set the delay low, we need to sweep the data from the start of the flash period, which is here at 12 minutes, up to the point where the CO2 is starting to go up. So right about there. Okay, so I've highlighted that data, and then if you go down here, you'll see our delta X, which is the time period, is 0.247 of a minute. That is the value that we enter under delay low, 0.247. Again, we can double click on that, 0.247. Okay, and our delay low is set. The next parameter is the delay high. Delay high is the point. Oh, and by the way, with the delay low, you can see that once it's set, the marker, which is a black dot on the graph, is what marks the start of the delay low, or the peak for integration. And as we've changed this parameter, the black dot lined up with it. So now we're setting up the delay high. delay high, you sweep the data again from beginning of the flash until the end of the CO2 peak here when the new steady state is reached, which is right about there. And again, you look at your delta x, which is 0.854 of a minute, and that is the parameter that you enter under delay high, 0.854. Okay, and you'll notice the green dot on the graph, which is our flag marker for the delay high point, has moved into position. So now we've got these two parameters set up, and the last parameter to set up is the event time. The event time is the period of time at the end of the flash and stop time over which data is averaged and is used in the interpolation of the reference levels. So event time should be set to about a quarter or a third of the flash time. So in this case, half a minute, with our flash time being 2 minutes, half a minute is going to be sufficient. It's basically looking at half a minute of the data at the end of that flash time to use in those calculations. So once all the parameters have been set, we can now proceed to the calculation page, which in this case is page 3 for stop flow. I'm going to shrink this window back down here. And if you go to your pages, you'll notice that there are 3 pages. Page 1 is raw data, page 2 is open auto, and page 3 is stop auto. The file calculates both open flow and stop flow data simultaneously. However, when the experiment is set up for stop flow, the open flow calculations are going to be meaningless. The oxygen differentials are just too small during the open flow portion of the of each time period to calculate VO2 from those differentials. So open auto, I'll just show you here in this data, is not going to be of any meaning for stop flow setup of experiments. So we go to page 3, which is stop flow, and on this is the page where we would be looking at our data. Here the VO2 and VCO2 are calculated from the diff CO2 flow and negative diff O2 flow. Those have been graphed here after they've been calculated. You'll notice that the oxygen peaks have been flipped to positive numbers, so integration can take place because you can't integrate negative numbers. You can zoom in at any point on this graph. You can stretch it, to see the data more clearly, and you can see that, these peaks are marked here with the delay low and delay high points on the graph. These user parameters are here as well for your reference. That it's the same user parameters that you've already set on page 1 of the raw data. It's the windows just show up on page 3 as well. And the VO2 and VCO2 as well as RQ are calculated for each of these individual peaks and plotted in the graph as well as shown in the table. You can move across to VCO2 stop and VO2 stop data to visualize. You have to remember though that there's only a single average number calculated for these parameters for the whole stop time. So each peak is integrated and one single average number is produced. So to see the actual numbers, you may have to scroll down quite a bit until you get to the point for each of the peaks. So in this case, VO2 stop is 2.32 and VCO2 stop is 2.53. So you can copy these numbers from the logger profile directly from this table into a spreadsheet program, or you can export the data by going to File, Export As, and select CSV, and save it to where you want to put this data. So I'll put it in here, and I'll just call this stop flow test. Save, and then you can then go to the place where you've saved it, open the file, and open it in Excel spreadsheet tab. You do want to convert, and then what you will see is you will see the whole data file. You're going to see all of the columns for all the parameters, all the raw parameters, all the individual sensors, as well as all the parameters for control of the file. So that's not something you want to look at. You probably don't want to see all the other calculations, but what you want is this VO2 and VCO2, and this data is going to be towards the end of this experimental spreadsheet file. That is the column right here for VO2 stop, and as I said before, you got to go and pull your numbers directly from each individual stop time peak, which will require some scrolling down. Here's the first peaks. Here's the first data from the peaks. And there are ways of sorting the data and eliminating all of the empty spaces as well. And that's information that's not going to be part of this video, but Qubit can provide a procedure on how to do this. Here's the next big data. So this is as far as we'll go with this particular file. So once you've sort of determined that these particular parameters are going to work with the setup that you have in place, then you can proceed to start your full experiment, with these parameters in place and collect your data. We recommend that if you're doing longer runs, that you save your data periodically to make sure that there's a power outage or some other situation that may result in software being stopped or hung up for whatever reason, that you don't lose your data. In this video, I will describe how to handle data from an open flow experiment. In this case, we used quite a large frog placed in the flow-through chamber that was connected to the Q Box, as I described earlier, for insect measurements. And as I said earlier as well, both stop flow and open flow calculations are done simultaneously in these files, experimental files, but the difference is in the setup of the user parameters. So again, flash time and stop time have to be set up before data is collected, and the flow rate through the chamber. And in open flow measurements, which are done on samples that are more active, larger animals like frogs, for example, where oxygen differentials are going to be large enough to detect during the open flow or flash phase, and they have to be at least 300 to 500 ppms, but larger differentials are going to be even better, and you can see that this is happening here. And one has to be careful that the CO2 differentials again don't exceed 2000 parts per million, which is the maximum range of the CO2 analyzer. In this case, what we've done is we actually used a CO2 free air to do the experiment with the frog, so there would be plenty of range available for the CO2 measurement. But as you can see, we're dealing with about 500 parts per million CO2 differentials and O2 differentials. You can see that on the dip CO2 and dip O2 graph as well. However, if you find that differentials are getting too close to 2000 ppm, the way to deal with it is by increasing the flow rate. The flow rate in this experiment was around 525 mils per minute, which is significantly higher than what we had for stop flow, and that is important in open flow experiments because the flow rate is inversely proportional to the CO2 and O2 differentials. So if the differentials are getting too big for the system, then one can increase the flow rate to reduce those differentials. And if the differentials are too low and getting close to detection limits, for example, of the oxygen analyzer, then one can reduce the flow to increase those differentials. It's important to note that the maximum flow rate for use with the Q Box RP1LP for the CO2 analyzer specifically should not exceed 650 mils per minute, as higher flow rates than that may pressurize the system and result in some damage. So maintain your flow rates lower than 650 if you're working with animals that require higher flow rates because they're very active. Then a second gas pump would have to be set up to subsample from the gas which leaves the chamber and deliver to the analyzers at a lower flow rate. So in the case of this experiment, we were measuring a single large frog. We had a flash time of 4 minutes, stop time of 1 minute, and we were getting good differentials here of CO2 and oxygen. So when doing open flow types of measurements, we usually recommend to increase your flash time because to increase the measuring time of the frog, and minimize the stop time, which should be as short as possible. During this time, the chamber is being bypassed to measure the reference level. It's important to get these reference level measurements, but they don't need to be very long. They can be as short as possible. All we need is just a reference. So as I said before, that the experimental file is exactly the same, and in the file, this time we're going to be looking at the open auto data, where the calculations of VO2 and VCO2 take place. And but before we go there, it's just as important here to line up the CO2 and O2 signals as we did with stop flow, and you do it in the same way. I'm just going to zoom in again to look at some data, couple of cycles close up, which makes it easier to determine whether the data is lined up or not. You go into your examine function again. Stretching this graph helps, and with the examine you select a couple of spots in the data where you have big enough in deflection or peak of some sort, because CO2 and O2 basically mimic oxygen inversely mimics CO2 responses. So you can see here that the data is pretty close. We probably don't need to worry about any delays here or lining it up. Maybe again we're looking at one or two sample data points. So I'll just adjust this by 2 and see whether that brings them. No, actually that made things worse. We may have to go reduce to a negative number, maybe -1. Let's have a look now at the examine, and yes, it was backing up a little bit, so the CO2 was being measured. Here we go. The two CO2 and O2 signals are now aligned with the O2 delay being set to -1. So once that's done, you can close this again, and we're bringing the graph back to its proper size. And we can go to page 2, where the open flow calculations take place. And again, those parameters have been brought over from page 1. And the things that we have to set up here is 2 parameters, event time and delay high. Event time of 1 minute is fine. Again, this is used in calculations, interpolation of the reference levels. And if you remember from the previous video, event time is usually about a quarter of flash time, so this is fine. 1 minute set up here. Back to page 2 again. The other parameter that we need to set up is delay high. In this case, it's the delay high is a flag which is being set up to show where the VCO2 and VO2 data is being calculated, because we're not gonna the data calculations at the beginning of the flash phase is going to be irrelevant, because at this point in time the chamber is being flashed, and during reference measurement of that 1 minute, a small accumulation of CO2 occurred in the chamber. So this needs to be flushed out before more steady state levels are achieved, which are representative of the open flow measurement. So this delay high parameter in the case of open flow basically sends a signal to the software where to start calculating the VO2 and VCO2 after this flash peak has gone through. And in the case of the data here, going back to page 2, we've got delay high set to half a minute. It's about half a minute to flash that peak, and you can see that, I'll just zoom in here, it basically means that the VO2 is not being calculated. Let's see, 20 is not being calculated for the first half a minute of the flash time, so there's no data here. But then in OpenFlow, the software calculates VO2 and VCO2 for every single point collected, every single data point collected, not like in StopFlow where a one average VO2 and VCO2 data point is calculated for the whole stop time. In the case of OpenFlow measurements, we have multiple data points that are being calculated and displayed in both the graph and in the software. And in the software, one has to find the VCO2 open columns where the data is calculated, and VO2 open columns. And those are the columns that are displayed on page 2 of OpenFlow calculations. And again, you'll see the data here can be either copied and pasted directly from logger profile into an Excel spreadsheet, as I explained before, or one can go to File, Export as CSV, and again save the data to a location, open flow test, and then once that's been saved, you can open that data directly in Excel and proceed to further analysis, whatever that may be. So here's the data, and again, going to where the open flow and stop flow columns are, to stretch the titles to see them more clearly. And there's BCO2 open, VO2 open, columns right here. And if you scroll down, you'll find the data for each flash time for every single point. So further analysis can be done from there. And as with any data after analysis has been done, it's a very good idea to save that as a brand new file with a new name so you don't override what you had there before, or raw data collected in case you make changes that you don't want to override. And you're ready to go. With these parameters set up correctly, you can then proceed to delete all data, clear all data here, and restart the experiment under a new file, but with those parameters in place.